Hello guys, this is Leonard Ang speaking, and I am one of the TAs for AMP, and I'll be going over Lab 8, which is over the digestion physiology. So the first thing that we're going to talk about is starch digestion through salivary amylase. And salivary amylase is an enzyme that is found in your mouth, and since your mouth is very neutral, it's the optimal pH would be 7 because 7 is a very neutral number. What salivary amylase does is that it breaks down starch, and what it does is that it breaks down starch and it goes to amylase hydrolysis, and then it turns it into sugars. So one thing that you did in your lab was that you had synthetic saliva and you had one test tube that you boiled it while the other one you left unboiled. And what boiling does to the enzyme is that it denatures it, and that means that no digestion occurs. So that means pretty much that means that the enzyme is now prevented from digesting the starch. Next is we're gonna go over the specific solution that we used to measure salivary amylase, which was IKI solution, or iodine potassium iodide. So when starch was present, the color would be a blue or a black color. However, if starch is absent, the color would be a yellowish color. So comparing your boiled saliva versus your unboiled saliva, you would notice that the boiled saliva, the IKI would stay blue-black the entire time. Well, that's supposed to happen. It might not have happened in your specific experiment during lab, but that's the ideal. So the IKI would stay blue-black because starch was not going to be digested because the salivary amylase is denatured. While for unboiled saliva, the IKI would turn from blue-black into a yellowish color, and that's because the amylase is now breaking that starch into sugars. So that's pretty much the basis for that specific experiment. Next, I'm going to go over the PhysioEx exercises. So the first thing that you did is that you measured the activity of lipase, and lipase is an enzyme that is found in the duodenum. So the duodenum is also neutral, so lipase's optimal pH is 7, meaning that at pH of 7 it's the most active. And what lipase does is that it breaks down lipids or fats into fatty acids through hydrolysis. And there's a thing called bile, and what bile does is that it's called an emulsifying agent, and an emulsifying agent is pretty much a chemical that helps facilitate the digestion of the lipid, so it helps the lipase out. And what you would notice is that when bile is present, the lipase activity would increase, and that is shown through optical density. So if there is an optic density of 0, that means that there's no lipase activity. However, any number above that would mean that lipase activity occurred and that the lipids was broken down into fatty acids, and most likely if there's bile present, the number would be a lot higher than without it. Next is for pepsin, and pepsin is found in the stomach and is active at an optimal pH of 2 because the stomach is very acidic because it has a lot of acids that need to break down the food down even further. So what pepsin does is that it breaks down proteins and it goes through hydrolysis and it turns it into amino acids and peptides. And you would notice is that when the optical density increased, the pepsin activity also increased. So higher the pepsin activity, the higher the number above 0 the optical density would be. And so those are the two PhysioEx exercises that we went over. It's very smart to go over it yourself and test it over to make sure that you got everything. This is just a general basis of information that you might need to know, because there might be additional specific information that you might find through your PhysioEx. Next is about your permeable dialysis, and this is dealing with absorption in the small intestines. So what happens is that you added a certain amount of sugar and a certain amount of starch into dialysis tubing. And what you did is that you tied that tubing and then you put it into a beaker of water. And what you would notice over time is that a certain molecule would exit out, would diffuse into the surrounding water, while the other one wouldn't. And so you had two solutions, which was Benedict solution and IKI solution. And Benedict solution tests for sugar, and what you would notice is that if sugar was present, the color would be red or a dark yellow. But if the Benedict solution was not, if the Benedict solution was a light blue, that means that sugar wasn't present. For IKI, which tests for starch, if starch was present, the color would be a blue-black color. However, if there is no starch found in the solution that you're testing, the color would be bright yellow. So what you would supposed to notice is that the sugar levels in the surrounding water was present. So the color was a red or a dark yellow. However, for IKI solution, there was no starch found in the surrounding water, so as a result, the color was a bright yellow. And so that means that sugar is more permeable, or sugar has an easier time diffusing through the membrane of the dialysis than starch, because sugar is a lot smaller molecule than starch, and starch needs to break down into sugars in order for it to diffuse through. So that's why it is important for your enzymes to break down that starch and bring it into sugars, so that it can use that and it can bring it through the cardiovascular system to use for nutrients. And that is pretty much it for this lab. And hope you do well.